This is a production of Cornell University.
So basically, just as Jenna mentioned, this is still a work in progress. However, I've had the chance to work with it for a little bit. So I hope to show some progress over what I showed last year, which at the same time creates a particular issue of my having to define what I already done to show what I have to do. But I hope I don't do such a bad job. So first is like, okay, so I'm going to talk about a little bit on what the project actually is, some of what I want to achieve with it, and what results I'm looking forward to get, and how I'm going to try and do that. So as background, all this project is thanks to CIMMYT data, basically. They have these germplasm collections, part of the Seeds of Discovery project, which includes about 4,700 maize landraces from all over Latin America. And the particularity of these landraces is that, well, they've been selected over by multiple generations where the breeders on these locations have been able to like keep them in location. So these landraces have been genotyped through GBS markers, which gave us about like 300,000 actually imputed markers. And the interesting bit of these landraces is that about 3,000 of them have geographical information data from where they were collected. These data includes these variables right here, which is basically environmental information that in theory has actually held some pressure on the way the breeders have been selecting the specific seeds that they keep for each successive generation to keep growing on their plots. So for example, we can think of precipitation. This is just a map showing where all these landraces have been collected from. And this is showing the normalized values for the precipitation variable specifically that has been averaged for the 6-month growing period. So we can see that depending on these landraces that have been growing, they have a tendency to have lower or higher levels of precipitation. Considering that maize is an outcrossing species, it can actually like just cross within the fields, and then the farmer selects whatever is growing better and then keeps the seed and then grows it again successfully over like however many years, because this collection has actually been kept in CIMMYT all the way from like 1950. So it's actually what we want to do with this specific data that we have is sort of like the typical association studies to hopefully identify alleles that are kind of like related, or not kind of, hopefully related to the individual variables that we'll be studying. So normally we think phenotype and genotype, we do an association study like GWAS or genome-wide prediction, and that could give us in theory alleles that have actually been associated to control this variable. However, as we might know as well, generally we don't have a full sequence genome for all of these landraces or whatever extensions we might be working. So they have to go through a process of imputation, which is like done thanks to like having some form of shallow sequencing, or in our case, GBS markers. So this imputation process has some issues, and I'll talk about a little bit. And as well, the association studies are compounded by problems such as population structure and the genetic architecture of the trait. This causes issues where like you might find identification of association with alleles that have nothing to do with the trait, just on the way in which the population has been kept and growing. So we have to control for that. And finally, those beneficial alleles that we're in theory identifying have also have to be validated, because, well, just the fact that we find it associated doesn't mean that they're actually causal for them. So my the overall objective of the project is kind of like tackle these 3 individual points to hopefully like just kind of leverage with the information that we have on these landraces that have naturally been selected to perform under these conditions. So thus far, what have I done? So the imputation kind of works. That means that we have a good sort of correlation, which is like a 0.67, which is not great. However, it's useful. However, as we can see on this plot, the imputation tends to fail over mostly on the minor allele frequencies, which tends to be the one that tend to have like a higher effect on the individual traits that we might have an interest on. So we have to improve that. This is done through Beagle because so far has been the best one that has worked for our specific material, because that's also one of the problems with imputation. It's totally dependent on the population that you're working with. The same sense, doing some association studies, just GWAS, particularly trying to explain the environmental variables, we have to include some kind of variable that's going to control for the population structure, because otherwise, what we tend to find is just basically these Manhattan plots where we have like incredibly significant sites all over the genome. So the significant bar is right here. So we have almost all of the thing being significant, but that's just because, well, the genome has been selected for the population structure in itself. So that's not great. So what do? We try different models. For example, a mixed linear model in which we include PCs and geographical covariates such as latitude and longitude. And what happens, we basically lose all signal. However, though, the most significant sites still tend to be the same, but they're not significant anymore. So what do we have to do? We have to basically find a way to basically increase the signal after control for population structure. So how do we do that? So the first step is basically, well, what happens if we just make a better imputation so we have better initial data? So for that purpose, the lab, the Butler lab that we are working on, they're trying to develop this practical haplotype graph, which is basically trying to use data from sequence assemblies, sequence genomes from maize, a diverse set of them, or just a simple high-depth sequencing, where basically they're trying to process the different assemblies, identifying k-mers over what are the specific, we're calling them reference intervals, which are in general just genic regions, because those are the ones that are simpler to impute. They include the promoter sequences and other mostly informative pieces of the genome, but at the same time, we're keeping the rest of it as well. So, however, this is where we're calling these haplotypes, and this generates a database of hopefully good curated haplotype structures based on the population that the database was actually generated from. So having those sequences that we already have, there's like 20 some assemblies as it is right now. What I would like to do is basically use this GBS data, or if we need some more data, pass it to the database to basically impute these land races into these other sequence genomes that are better assembled. So hopefully that will give us the possibility to identify associations more likely, or with better, with higher power. Why? Because, well, instead of, if we're trying to identify association between SNPs, we can have like hundreds of thousands of them, even millions, which basically reduces the ability to identify association because you have so many things to test for. If instead of testing for hundreds of thousands of them, we're just identifying several tens of thousands, which are just the gene regions, we might get a bump on that. In the same way, by we have also these land races already sequenced, some of them. So that might also give us the ability to test this imputation approach to see if it's actually working. But again, this might require more so that we'll see what happens with that. And as it is a PhD, the practical haplotype graph is being developed, so it's going to be part of the project in its own. The other thing is to the association approach. So once we have these imputed genotypes, there's these approaches that are using summary statistics from GWAS, where we're actually just using the end results for them. And what it actually does is sort of using the genetic structure of the individual traits that we're looking for to find association. And basically the fact that the population structure in its own is sort of shared, so that you're hoping to share alleles in there, and also the genetic architecture for the individual traits might be shared as well. This specific approach is called MTAG. Looks actually to identify a bump on the association by just mixing that up. And at the same time, if we wanted to develop this data to be stored on a database, keeping the individual data is kind of, there's a lot of information. So using summary statistics might actually allow us to keep the data accessible for more people. Another step, sorry, is to actually, instead of using just the individual genotypes that we were doing, is to try and do the association through these haplotypes by potentially allowing I'm sorry, I'm losing my voice. The alleles that are on their higher frequency, we could potentially impute the individual alleles between them that are on lower frequency, which cannot be genotyped by a shallower sequencing approach. But of course, it's completely dependent on the fact that we might not get good haplotyping. But again, this is stuff that needs to be tested. And finally, all of these association studies through the environmental variables, how can we test that? Well, we have some information for flowering time trials for these land races that have been found on all these different regions across Mexico that have been selected to be like sort of performing well under the individual environments that they are there. So I tried some individual genome-wide prediction models, and we can see that both using the environment as a predictor for the flowering time data is giving us some kind of signal, which is telling us that the variables are related to the way that these land races can perform in there by themselves. However, of course, using the normal genotype approach as predictors for these flowering time data, we have a sort of like relatively good prediction of 0.5 correlation between days 2 and thesis over these multiple trials. So what we would like to see is basically if we select different genotypes or these regions for the genome, we should be able to see an improvement over this prediction, or at least that's what I would like to test for. And I'm gonna just skip on this. Sorry about that. So I want to thank the people at CIMI for all the data and the support, and definitely the Buckner Lab, because all of this is thanks to them and the research that everyone is doing and we're sharing. And I'm gonna see it for my scholarship to one over here as well. So if you guys have questions.
Any questions for Arcadio?
I'm curious about your environmental data. Are you looking at like an average over a certain historical time period, or as singles, or rather, are you taking into account variation as another sort of layer on top of that?
Right, so we have the variables individually for each of them, the months of the year. But the stuff that I've been working with has been averaged over the 6-month growing period for the field season. So I could definitely test the individual months and see how they do. Thus far, the 6-month growing period has seemed to be working pretty well.
That's 6 months in a single year?
Yes. Yeah, so this is all data that's kind of like average for the location on which each of these land races have been growing. And that's sort of like the number that's understood to be occurring in that location. Yeah.
Have you looked at prediction, genomic prediction, or maybe associations, looking only at the genes, at the genic regions, given that you're going to be later relying on imputation only around the gene regions?
No, I have not. And that's basically what I still want to do. So what I've done is just genomic prediction across the whole genome. And that's basically what I'm interested in, is sort of furthering down to just these genic regions. And then hopefully the prioritization that I can get from the GWAS studies to make that sort of like hopefully increase prediction accuracy. But I have not tested just on the genes. However, since I have like GBS data, it might already be sort of biased towards that. But after imputing, I'm not sure. Yeah.
All right, let's give Arcadio another round of applause.
This has been a production of Cornell University, on the web at cornell.edu.